So we're going to start moving into what are called macromolecules. These are the large biologically active molecules that exist in cells. And really we're going to talk about two types. One are the proteins, which we'll talk about first. It'll take a few lectures. There's a lot to say. They're really complicated, and we'll spend a little bit more time on those. And then we'll go into the nucleic acids, which of course is DNA and RNA. And that won't take as much time because they're not as complicated and versatile as the proteins. So as I think I mentioned before, the amino acids are, the macromolecules are made up of polymers, basically small subunits linked together. With the nucleic acids, the polymers are comprised of nucleotides. These are the subunits. And with the proteins, which we'll start talking about now, the subunits are the amino acids. And there's 20 different amino acids, different properties. And let's just take a look at kind of one prototype amino acid that's right in the middle. I have under amino acids written in green. And the amino acid, all the amino acids basically have three parts that I want to draw your attention to. On the right-hand side is what's called a carboxylic acid group, C double bond OOH. On the left-hand side is an amino group, NH2. That's why they're called amino acids, because the C double bond O, it's actually O minus, exists in cells. So it's acidic, so they're amino acids. And then we have the central carbon, which is called the alpha carbon. And off of the alpha carbon is what's called an R group. And there are 20 different R groups that exist. Actually, there's a couple more. There's 22. But for the most part, there's 20 R groups that exist. And they have different properties, which we'll look at in a little bit. But they all have that same structure, with the R group in the center of the amino acid. Now, often we talk about proteins in terms of their size. And when we talk about the size, we're talking about the number of amino acids in a protein, and they range. An average size protein I've written is around 300 amino acids. So if you were to go into a cell and just kind of randomly pick a protein out, chances are it would be around 300 amino acids or so. But they range. They range from extremely small, what we call peptides. 9 amino acids are just about the smallest active proteins that are known, and these are small peptide hormones. Vasopressin, oxytocin is another one. These are small, really tiny 9 amino acid small proteins that have activity. And then they can be, like I said, around 300 amino acids, but they can range all the way from that 9 amino acids. Myosin, which is a protein involved in muscle contraction, is around 2,000 amino acids, and titin, which is, I think, one of the largest proteins known, is 27,000 amino acids. Now, the proteins carry out an enormous number of activities in cells. They carry out digestion, they carry out construction, they carry out movement, they carry out sensing, they transfer signals, they are signals. It's basically all the activities that a cell carries out is done by proteins. There are some caveats to that, because as we're learning, RNA also has activities in the cell, but the proteins seem to be carrying out most of the basic biological activities in cells. Now, as we'll see as we go through this, the specific function of a protein is determined by its structure. Each protein that we look at has a different structure, and each protein has a different function. So the structure determines the function, and we'll look a lot at that in a little bit. The first thing I want to do is look at the amino acids. I want everybody to take a look at all these, and we're going to look at them carefully and kind of see what they look like. So let's go on to the next slide here. Okay, so these are the 20 amino acids used in protein synthesis. Hopefully it's clear on the slide. There are, in the textbook, there exist other figures, and you can find these, all over the internet. But these are the 20 amino acids, and let's just take a look. First of all, they're divided into really 4 groups. Nonpolar and polar, number 1. So again, we come back to that nonpolar and polar properties. That is, nonpolar is hydrophobic, does not mix with water. Polar is hydrophilic, it does mix with water. Then there is what are called electrically charged, which are all polar. Anything with a charge is polar. And we have what are called the acidic and basic amino acids. We'll talk about those in a second. But let's go up to the top and look at the first amino acid there, glycine. So you see on the right-hand side the carboxylic acid portion. You see on the left-hand side the NH3+, which is the amino portion. And the R groups in this diagram are facing downwards. So with glycine, which is the simplest amino acid, the R group is a hydrogen. If we move over one more to alanine, you see the carboxylic acid, the NH, the amino portion, and the R group is CH3. If you look at valine, the R group is CH, and it's split off, and there's 2 CH3s attached to it, and you go on and you can see that these things just get a little bit bigger, a little bit more complicated. If you go down to phenylalanine, there's basically a benzene ring-like structure. Now these are all nonpolar amino acids. It's not really the amino acid that's nonpolar. This category is referred to the R group. So these are all nonpolar R groups, and you can see they're all nonpolar because there's no oxygens, there's no nitrogens, there's no asymmetric charges going on here. They're mostly CH groups. There's a little bit of polarity with the sulfur, with methionine, but nothing very significant, so it's still considered a nonpolar amino acid. So all these are hydrophobic R groups. Now we look down at the polar R groups, and same kind of thing. You look at the first one, serine, CH2OH is the R group. So you have that oxygen with the hydrogen, so that makes it polar. Again, you look at threonine, it's similar to valine, except for it has an OH instead of a CH2 or CH3. Cysteine, tyrosine with an OH, asparagine with an oxygen, which is polar, and NH2. Another group on there. So these are all these are all polar amino acids or hydrophilic, meaning that these R groups will interact with water. The nonpolar R groups will not interact with water. And it's going to make a huge difference on how these things participate in creating the shape or structure of the protein. Now we look at the electrically charged, and there's 2 acidic, the C double bond O minus, and then you have glutamic acid, which is CH2CH2, and the C double bond O minus. All acids give off their proton at neutral pH. So that's why they're acidic, they give off the protons. Bases accept protons at neutral pH. So the 3 on the right-hand side, lysine, arginine, histidine, you can see there's NH groups there, and all those are positively charged because they accept protons. So the acidic and basic are fully charged. Also, they're polar because they can interact with water, that positive charge. Now, the thing about the acidic and the basic amino acids is that they can change their properties as a result of pH. And there's a value called pK, which is kind of illustrated at the bottom, which gives an indication of the acidity or the basic properties of the R groups. So aspartic acid has a pK of 3.9. Glutamic acid has a R group, a pK 4.2, and in a little bit I'll describe that in more detail it'll make more sense, but just know that these can change their properties as a function of the pH, and thus proteins with these amino acids can change their properties or their function as a function of pH. Now, people always ask me at this point, there's always hands going up, and I'm assuming there would be hands going up if we were meeting face to face, and that is, do you need to memorize these? And the answer is no, you don't need to memorize these structures, but you do need to be able to recognize them and their properties. So you'll need to be able to recognize which amino acid R groups are nonpolar, which are polar, which are acidic, which are basic, and so on. So you don't have to memorize them, but you do have to be able to recognize what their structures are and what their properties are. Now, the other thing I want to say about amino acids is that, of course, they are absolutely required for the construction of proteins, but they also play other roles in metabolism, not only construction of proteins. And I want to give you a really interesting example on the next slide. Okay, one last thing that I want to bring your attention to in terms of the amino acids is that I have an asterisk written here, and I have a number of amino acids that are marked as essential amino acids: valine, leucine, isoleucine, methionine, tryptophan, phenylalanine, threonine, lysine, and histidine are all essential amino acids. Does anybody out there know what an essential amino acid is? Too bad you can't answer in this format, but I'm guessing that at least some people don't know what essential amino acids are. But basically, the essential amino acids are amino acids that we need to get from our diet. All the other ones we can synthesize. We can synthesize glycine, alanine, serine, and all those other amino acids. Essential amino acids we need to get from our diet. And that becomes important, so we have to eat right. And often, if somebody is a vegetarian, you can get all the amino acids that we need from meat, chicken, dairy products, I think eggs. But often you'll hear that if somebody's a vegetarian and they're not eating meat or they're vegan, you need to make sure you're getting the proper amino acids in your diet. And so sometimes you have to make sure you eat the right complement of foods so that you get all the various amino acids. If, for example, you don't get leucine in your diet, then any protein that requires leucine will not be made. And so you get you can get these pretty severe dietary deficiencies. So essential amino acids are those that we need to get from our diet. So here's an example of tryptophan. Now you remember, we just mentioned tryptophan is one of the essential amino acids, meaning that the only way you can get tryptophan is through your diet. We don't synthesize tryptophan like we do the other non-essential amino acids. And as it turns out, tryptophan is the direct precursor for two neurotransmitters, serotonin and melatonin. So there are biochemical pathways. Although tryptophan is used in protein synthesis, there are biochemical pathways that convert tryptophan into serotonin, and a biochemical pathway that converts serotonin into melatonin. Now, what are these serotonin and melatonin? Well, most people have heard of them. Serotonin is a neurotransmitter that appears to be involved in mood. And people with very low levels of serotonin sometimes can exhibit signs of depression. And so some of the classic antidepressants are called serotonin reuptake inhibitors, and that is they they block the uptake of serotonin and its degradation, so they leave it in the system longer, so it increases the concentration. So serotonin is important for mood, and then melatonin is a transmitter that is involved in sleep cycles, and that the amount of melatonin increases in your blood and has an effect on brain as you go to sleep. Now, when I saw this, it occurred to me that, well, if tryptophan is a essential amino acid, and the only way we can get it through our diet, is it possible, now let's just talk about serotonin, is it possible that some forms or some individuals with depression may have low levels of serotonin because they simply don't have enough tryptophan in their system? You need to get it from your diet. What if you're not eating foods that have tryptophan, so you're not going to have the precursor for the serotonin? So I kind of did a little bit of research, and it turns out that that seems to be true. And that is, people who are depressed, or some people I should say, who have low levels of serotonin can increase their mood by changing their diet to foods that have more tryptophan. What foods have more tryptophan, if you're interested? I only know a few. Pumpkin seeds apparently have lots of tryptophan. Turkey has lots of tryptophan. In fact, that's why some people say that, well, maybe you get sleepy after you eat turkey, is because it increases melatonin. That may be. So there was pumpkin seeds, turkey meat, and also cheddar cheese, for some reason, has high levels of tryptophan. Okay, so just know that there are other important pathways that require amino acids. Now I want to talk a little bit about the acidic and basic amino acids, and that's next. So as I mentioned before, the acidic and basic amino acids can change their properties as a function of charge. And that charge is a function of the pH. That's the R groups. So let me describe this slowly, because sometimes it's a little bit hard to describe. But let's take a look at aspartic acid. Well, first of all, look at neutral pH. That's neutral pH is 7. So if you look at neutral pH, the acids of the R groups give off their protons. So to pH 7, they're charged. They give off the protons, so they're charged. So aspartic acid has a negative charge. Glutamic acid has a negative charge. As you increase the acidity, so on the left-hand side basically shows the pH. So 1 is extremely acidic, 13 is extremely basic. Now when you have a high acidic concentration, or excuse me, when it's high acidity, it means there are high concentration of protons in the solution. So as you go from 13 to 1 in the pH, the number of protons in the solution increases. So if you look at aspartic acid, for example, as you go from pH 7 and you start decreasing the pH, what happens is at around pH 3.9, the R group grabs the proton and loses its charge. So at 3.9, there's a conversion that takes place from charged to uncharged. So as you decrease the pH, increase the number of protons, at some point the concentration of protons is such that it goes back on. So by reducing the pH to 3.9 or lower, that R group becomes uncharged. The same is true for glutamic acid. That is, at neutral pH, it is charged, but as you decrease the pH, in this case to 4.2, it grabs a proton and becomes charged. So you can change the charges on these amino acid R groups by altering the pH. the pH at which it either accepts or loses a proton. So above 4.2, it loses the proton, glutamic acid. Above 3.9, it loses the proton, that's for aspartic acid. So that pH, where you get that switch, is referred to as the pK. Now, if we look at lysine and arginine, we're not going to look at histidine. Histidine is a little bit more complicated because it's a very, very weak base. So it's hard to really describe that clearly. But look at lysine and arginine. Lysine has a pK of 10.5. That means below 10.5, it accepts the proton. Just like with the acids, below 4.2 accepts the proton. So below 10.5, lysine accepts the proton. Below 12.5, arginine accepts the proton. So pH 7 is below the pK of both of those. So the basic amino acids are charged. They have an extra proton at neutral pH. But above that value, above 10.5, they lose the proton and become uncharged. Okay, so in all cases, below the pK, accept the proton. In the case of the acids, they become uncharged. In the case of the bases, they become charged. Above the pK, they lose a proton. In the case of the acids, they become charged, negative. And in the case of the bases, they become uncharged. They lose that positive charge. If you have questions on that, send me an email, or we can meet sometime, but that's something that's important. Now, these amino acids link together in what are called peptide bonds to make proteins. And here's the way it works. So here we have two amino acids. They're not showing the R groups for this first example. So at the top, on the top left, you have one amino acid with an R group, can be any. Next to it, plus, you have another amino acid with any R group. And what happens is, if you see that OH and the H on those two respective amino acids, the OH from the carboxylic acid portion and the H from the amino portion, what happens is that carbon and that nitrogen are going to link up with a new bond, covalent bond called a peptide bond, with the release of water, H2O. And so you can see there's the arrow with the release of water, and now you have these two amino acids linked. So there's the carbon with the double bonded oxygen, now linked with this peptide bond to the amino group, which now has one H, and then you go on to the next R group. So these are now linked with two amino acids in a small peptide. So you see below a small, what we call a peptide. Peptides are proteins which are tiny, small number of amino acids. But basically the proteins would be a long polymerization of these amino acids. So here we have a tripeptide, which has one R group, is phenylalanine. Then you see the peptide bond. Then you see the R group of cysteine. Then you get another peptide bond, and the third R group is valine. Now, there's a couple things I want to point out about this. First of all, there is a specific orientation that we pay attention to, kind of a directionality to the way this thing works. So on the left-hand side, you have the NH3. That's classically how they're drawn, with the NH3, which is the amino portion, on the left-hand side, and the carboxylic acid portion on the right-hand side. So that's referred to as the amino terminus or the N terminus, or the carboxy terminus or the C terminus. And we use that nomenclature quite a bit. So we may be talking about some particular protein that's, let's say, an enzyme, And we may say, oh, the mutation that destroys that enzyme activity is near the carboxy terminus. It's just a way for us to say kind of generally where it is. Or there's a linkage of some other compound to the amino terminus. So it just gives you an idea of the orientation and where these things might be. Now imagine that this, and the other thing I wanted to mention is that anytime a new amino acid is added, it's always added to the carboxylic acid or the carboxy terminus. So if we were to add cysteine to this, or we were to add, proline or whatever amino acid next, it would be adding on to that C-double-bond-O side. That's the directionality of the synthesis. So here's another drawing that just kind of shows a similar thing. This has 4 R groups. You see the amino terminus, a carboxy terminus, what they call the peptide backbone with the peptide bonds illustrated. And there are 4 R groups, or sometimes called side chains: histidine, aspartic acid, notice that it's charged, leucine, and tyrosine. Now, we're going to talk about this a lot, and that is the sequence of the amino acids in a protein. Let's say it's a 300 amino acid protein or 100 amino acid protein. The sequence of the amino acids in a protein gives rise to its specific function. So imagine we have 4 here, and imagine it extending from the carboxy terminus, adding more and more. Imagine that there's 100 of these things. 100 of these R groups. They're not very complicated. Each R group in its own is not very complicated, but when you put them all together, you get a very complicated assortment of chemistries there. Now, it turns out that we may have in our bodies, it's not really clear, people are looking at it, but maybe it's possible we have 500,000 proteins, maybe more. Say there's 500,000 proteins. Now what I want to describe to you is that with the 20 amino acids, in the way they can be linked up together randomly, there's an enormous amount of chemical diversity. A sequence of 100 amino acids, it has a chemistry to it, an activity. A different sequence of 100 amino acids has a different chemistry, different activity. A third protein with 100 amino acids has a third very complicated chemistry and function. So there's an enormous amount of diversity available to us. We use maybe around 500,000. In the next slide, I have 150,000, but it's anywhere between 150,000 and 500,000 proteins. Now let's just take a look at the next slide, and I'll try and describe what I'm trying to get at here. So the sequence of amino acids in a protein, and I'll talk about this again in the next slide, but the sequence of amino acid in a protein is referred to as the primary sequence of a protein, or the primary structure of the protein. The sequence is the primary structure. Now that primary structure can differ, because that depends on which amino acids are there. It's basically the amino acid sequence. So what I have written here is an enormous amount of diversity and potential. So we have 20 amino acids. Each amino acid can be linked in a chain. Each amino, the next one in the chain could be any one of 20, and the next one in the chain could be any one of 20, and the next one in the chain could be any one of 20. So if we have a 4 amino acid peptide, The first position could be 1 of 20, second position could be 1 of 20, third position 1 of 20, fourth position 1 of 20, which equals to 160,000 possible combinations. 160,000 possible combinations from just 4 amino acids. And so the equation actually is 20 to the nth power, where n is the number of amino acids in the protein. So if we had, for an example, an average protein of 300 amino acids, how many possible combinations would there be? There'd be 20 to the 300th, which equals 10 to the 390 possible structures. That's 10 with 390 zeros coming after it. That is basically an infinitely large number. 10 with 390 zeros after it. Now, to make a person, I've written here, it varies, the estimates vary. I've written here 150,000 proteins can make a person. It may be as high as 500,000. So that's 10 to the 5th proteins. 10 to the 5th proteins. If it's 500,000, it would be 5 times 10 to the 5th, 5 zeros. Whereas the number of possible structures of 300 is 10 with 390 zeros after it. So the point is that with the 20 amino acids, we can have an enormous number of possibilities, an enormous number of functions, and we are only using a small, tiny, minuscule subset of those functions to make a person. And one of the goals of nanotechnology is to try and design new functions that maybe haven't been seen before, new activities, by combining these amino acids in different ways, because anything basically is possible. As long as it's thermodynamically possible, a protein can be made to do it. So there are multiple levels of protein structure. The sequence of amino acids is the primary structure. So there's multiple levels, and as I said, we're going to talk more about this later, but I just want to tell you what they are. So the primary structure, this is the sequence of amino acids linked together. It's the simplest thing, although it's responsible for the function. The secondary structure represents kind of small regions of folding, tiny little regions, and we'll look at those. Tertiary structure is larger, more complex regions of folding, larger areas. And then the quaternary structure represents situations where we have more than one protein associating into a much larger structure, like some of the ATP synthase has multiple proteins, or the proton pumps that are used during electron transport have multiple proteins. So those all have quaternary structure. So we'll talk about each one of these a little bit later. But I want to go back again to the primary structure and its importance. So here we have the primary structure of a small peptide, and imagine this thing is 100 in length, or 300 in length, or 600 in length. And so you have all these R groups coming off of the protein, and the R groups interact with themselves, they interact with water, and the bonds are flexible. That is, there's lots of rotation and flexibility in these things. So the R groups can spin around like horizontally in the plane of the figure. They can spin vertically. They can fold. They can bend. So these things can make all kinds of folds and bends. And so from any single primary structure, from any sequence, there's an infinite number of ways these things can fold up. An infinite way. It's like the diversity in the types of structures that can be formed, in principle, are enormous. But what happens is it only folds up into one. Only one structure. It's a little bit of exaggeration. Sometimes you can have more, you can have 2 or 3, but for the most part it's very few. But you can have it folds up into a specific structure that is dictated by the primary sequence. And so you end up getting very specific structures and very specific functions. And this shows kind of a diversity in the types of shapes that proteins can fold into. And there's an enormous number of shapes. But again, only one of those shapes gives rise to the specific function. So this kind of gives an illustration, we're going to look at more later, of the types of structures that proteins can fold into different proteins. So you have catalase, folds in, has a primary sequence, allows it to fold into that structure. Lysozyme has a primary sequence that allows it to fold into the structure that's shown there. Alkyl dehydrogenase at the bottom, specific primary structure folds into that. Well, what we now know is that the protein structure, or excuse me, the primary sequence, that is the sequence of amino acids, is the only determinant of the three-dimensional structure of a protein. That is, the primary sequence dictates the proper way this thing should fold up, and the foldings get very complex. So let me show you an example of a classic experiment that was done a number of years ago. An individual named Christian Anfinsen, who was working for many years on how proteins fold. This was again in the 1960s. How proteins fold. What are the forces that control protein structure? He ended up getting a Nobel Prize, not just for the experiment that I'm going to show you, but for much of his work leading to our understanding of complex protein structures. So here's an experiment, and what it says is, first of all, the conclusion: denatured proteins can often recover their natural shapes. Denatured protein is a protein that loses its three-dimensional structure, and that can be done by heat, or it can be done by compounds like urea, which are highly charged and they mess up some of the bonding. And then it says this type of experiment demonstrates the conformation of a protein is determined solely by its amino acid sequence. So here's the experiment. He used a protein, an enzyme, which is called RNase. It's a tiny protein. It's 124 amino acids. Now, if the way he measured this is if the RNase is active, then the assumption is it's in the proper conformation, the proper shape. So if it's in the proper shape, if you take this purified RNase and add an RNA molecule to it, it will digest the RNA molecule. So that's how he was able to determine if the thing is functional or not, and assumption that if it's functional, it's in the right shape. So on the left-hand side, he had the purified active protein. He added a high concentration of urea. He also did it by heat, but in this case it's showing urea. And what urea does, it causes the protein to denature. That is, it unfolds and it becomes randomly, kind of randomly floating around. The primary sequence has not changed. Still the same amino acid sequence. Now, if you slowly remove the urea, what he found was that the enzyme became active again, indicating that it had folded into the proper structure. So his conclusion is that the only thing that's required, now this was done in water, by the way, in pure water, to which he added urea and then he removed urea. So the conclusion is, and this is kind of gets back to what I was talking about when I talked about the hydrophobic and hydrophilic properties, that they give rise to complex structures, here we have an example of a protein with a primary sequence, and that's all that's required. That's all the information that's required for this protein to fold up correctly and carry out its activity in the presence of water. So it's the primary sequence. It's the arrangement of those amino acids somehow allows this thing to fold. There's no other information. There's no RNA in there. There's no DNA information. There's no other proteins. There's no energy, no ATP. It just spontaneously folds up. And that's due to the sequence of the amino acids. So let's take a look at some of the forces that are required to determine correct amino acid, correct protein structure. Okay, so we know that if we take a protein, long stretch of amino acids, and throw it into water, it will spontaneously fold up into the correct structure. So what are some of the forces that determine that correct structure? So there's 5 that I want to mention. One is hydrophobic and hydrophilic interactions of R groups with water. We'll look at that very briefly, but I don't want to talk about that too much. We would understand that the polar R groups would interact with water and the nonpolar would move away from water. So that would create some kind of three-dimensional structure. There are bonds or interactions between the R groups. There are interactions between nearby peptide bonds. We'll look at that. Then I want to mention proline because it has an interesting structure and it often will create bends in a protein. And then there's what are called cofactors, such as magnesium or zinc, which participate in how these things fold. Now, before I talk about that, I just want to, I have written in here, just looking at, for example, bonds or associations between R groups. I just want to mention one in particular, that there's a particular amino acid, which is cysteine, which when they're brought together, 2 cysteines are brought together, they can form a covalent bond. And that's what's shown here. I'm not showing the whole amino acid, I'm only showing the R group, so CH2SH. And then coming right next to it is the SHCO2, CH2, which are 2 cysteines brought together. And under that condition, there's a reduction reaction that takes place. The protons come off, and the 2 sulfurs are linked together through what's called a disulfide bond. These are very strong covalent bonds and participate, I'll show you later, participate in the interactions between proteins or among proteins or within a protein, and they form very stable bonds that are very difficult to break. But we'll talk about that in a minute. I just wanted you to know there was only one type of covalent bond between R groups, and that's the disulfide bond. Just go back to number 1 for a second, the hydrophobic-hydrophilic interactions. And this kind of demonstrates in a very simple way how this would work. So here you have an unfolded polypeptide, an unfolded protein, and each one of those circles is supposed to represent a different R group. Some of the R groups are polar, some of the R groups are nonpolar, that is hydrophilic or hydrophobic, and you put that into water. Let's say it's the RNase. You put that into the water, and the hydrophilic side chains hydrogen bond with water, we saw that, and the hydrophobic will move away from water into the center of the protein. So what this is showing is that the position of these different polar and nonpolar R groups participate in how these things fold up into a more complicated structure. And they show the folded conformation in an aqueous or water-based solution. That's simply due to the arrangement of polar and nonpolar where they're placed. Now let's look at interactions between R groups. Okay, so here I'm showing just a line diagram of a protein. The amino terminus is labeled number 1. The carboxy terminus is a 400 amino acid protein. The carboxy terminus is labeled number 400, so there's 400 amino acids in this protein. And we're really only looking at 6, but realize that there's 400, but we're only looking at 6. So we're looking at lysine at position 20. We're looking at serine at position 141. We're looking at cysteine at 184. We're looking at cysteine at 220. Glutamine at 266. And glutamic acid at 325. Now there's a whole bunch of things I want to talk about here. First of all, let's look at the different interactions. So at the top we have the disulfide bond between 2 cysteines. That's what I showed you earlier. So at 184, at 220, these 2 cysteines come together and they form this disulfide bond, covalent linkage. Now we look at serine 141 and glutamine 266. These things are brought close enough together so that they can form a hydrogen bond. So that's another type of interaction between an R group. They form a hydrogen bond because you have the double bond O, which is partially negative, and you have the hydrogen coming off of that oxygen, which is partially positive, like we've seen before, and so you get a hydrogen bond between those. And then finally, we look at lysine at position 20 and glutamic acid at 325. And these things are also brought close together. And in this case, you have a basic amino acid in lysine, acidic amino acid in glutamic acid. So you have 2 complete full charges, a positive and a negative. And so those undergo an ionic bond. So we have a covalent bond, we have a hydrogen bond, and an ionic bond. These are the types of interactions that can occur between R groups. Now, another point. That is, I wrote along there the pK. So the pK of lysine is 10, the pK of glutamic acid is 4.7. And so my question is, what would happen, knowing what I just told you about the pK, what would happen if we were to drop the drop the pH to pH 3, let's say. Drop the pH to pH 3. So what would happen would be that glutamic acid would now accept the proton. So you would lose that positive charge. The negative charge. You lose that negative charge, and that ionic interaction can no longer occur, and so those amino acids come apart. So by changing the pH, you can alter the way these R groups interact very easily. Now here's the other thing. And that is that these amino acids that I'm showing you are a long distance apart. They're very far apart on the primary structure. Yet the folding brings them right next to each other. So the three-dimensional folding of these proteins brings the amino acids, or I should say it maybe a little bit differently, the interactions of these amino acids creates the three-dimensional folding that occurs, bringing R groups that are far away from the protein close together in order to interact. Now, there are 400 amino acids total in this protein, and we're only showing 6. What I want you to try and envision is that there's all these hundreds of interactions taking place on this protein, creating a very complex structure that would occur due to all these interactions with all these R groups. So even though amino acids are far apart, it doesn't matter, because the folding brings them together in order to act properly. Finally, the precise position of these amino acids on the structure is what allows these things to interact. So we have serine at 141 and we have glutamine at 266. If the serine, and they have to be there in order to interact. If there wasn't a serine there, of course it wouldn't interact. You wouldn't get that structure. If the serine was at 143, you wouldn't get the interaction. If the glutamine was at 267, you wouldn't get that interaction. So the precise positioning of these things is crucial. It's absolutely crucial. And so that's why we say that the primary sequence, the precise position of each amino acid on this thing, is responsible for giving rise to the proper three-dimensional structure, because these things need to be there in order to get the right interaction. So that's why, for example, many diseases that we're aware of, particularly inherited diseases, even cancers, are due to single amino acid changes. Cystic fibrosis is due to a single amino acid change in a protein. Sickle cell anemia is due to a single amino acid change in the protein, because removing the single amino acid or changing that amino acid results in a specific interaction not occurring. And when that specific interaction does not occur, the protein cannot fold properly. So these things are crucial in how these things fold. Let's look at the next slide. This one basically just shows peptide bond interactions. As I mentioned, that you can have the R group interactions with water, the interactions with the R groups, among themselves, like we just saw, and also peptide bond interactions. So here, let me describe what's happening. So on the top, you have a length of a protein. I'm just showing the R groups as R groups, but you see the peptide bonds, the C double bond O N H, and then another R group, C double bond O N H, and then another R group. That's the peptide bond. So on the top you have one length, on the bottom you have another length of a particular protein. And what happens is, if these things are brought into the proper position, then you can get hydrogen bonding between the peptide bonds. You can see that the double bonded oxygen will hydrogen bond with the hydrogen coming off of the nitrogen. So if you were to get a whole length of these things happening, like a whole run of them, it can lead, depending on the R groups, it can lead to different effects. You can get stacking, or what are called sheets, beta sheets. We'll show you those later. And you can kind of see how that would happen. These things would just align up and kind of stabilize themselves. You end up getting these sheets. Or you can get a coiling effect, which turns out to be called what's an alpha helix, which we'll also look at later. But the point is, through these peptide bond interactions, it can stabilize the protein and create certain structures. Finally, let me just tell you about proline, because it's an unusual amino acid and it causes some kind of kinks in proteins. So here we have, I'm showing you two amino acids, valine and proline. Let's look at valine. You have the amino terminus, carboxy terminus, and then you have the R group with the CH3, and then CH3, CH3. Actually, that should be a CH2 in the center. CH3, CH3. I'll change that next time. And that, as I mentioned, these R groups can rotate. They can rotate around. They have free rotation. Now, if you look at proline, proline is different. You see the carboxy end of the amino acid. You see the R group coming off of the central carbon. But what happens is you have the end of the R group making a covalent bond back to the nitrogen. So it makes a ring-like structure, which in which there's a loss of rotation. The R group cannot rotate, and it forces a bend in the protein. So proteins have lots of proteins have lots of little sharp bends in them. So in addition to all the other interactions, the positions of proteins are extremely important. So the bottom line here is that the primary structure, so you have these proteins that fold up into very complex shapes, and we're going to look at some of those in more detail. The complex shape is determined by the primary sequence, the sequence of amino acids, and that, of course, is determined by the sequence of the DNA, which encodes the protein. So the sequence of amino acids is crucial and drives the three-dimensional structure in the presence of water. And all of these interactions that take place among all these amino acids are very specific. And the precise positioning is very specific. And if any of that's changed, then of course you get a different structure, you get a different function. So the net result is that any mutation or any change in the structure, any mutation that would change any amino acid can lead to drastic changes in the structure of the protein. Very specific three-dimensional structures are required. Okay, we will stop here. I didn't mention the cofactors. I'll mention those another time. But we will end here and talk more about structure next time.